सर हमने उनके ड्रॉप पे कुछ चीज़ उनके मुझे काम कर बिस्मिल्लाहिर्रहमानिर्रहीम बिस्मिल्लाहिर्रहमानिर्रहीम Good afternoon. I welcome the Vice Chancellor and my colleagues and research students in the third seminar of the DSA Events Calendar 2021. Today, the seminar is about the application of genomics for livestock improvement in Pakistan. Dr. Bilal will give the seminar. It is about 40 minutes, and then we have the question and answer session. So I request Dr. Bilal to start the speech.
Bismillahirrahmanirrahim. I am thankful to Director of Advanced Studies for inviting me for this lecture and sharing my knowledge. So our topic is application of genomics for livestock improvement of Pakistan. So genomics is a new technology, and today we will discuss how we can implement and use this technology in Pakistan for improvement of local breeds of Pakistan. So why genetic improvement of local animals? It's a common slogan that high producing animals are available in the world. For example, Austrian Friesian and beef, world-renowned beef breeds are available. So why should we go for local animals? Many industrialists, they think that we should simply import foreign animals in Pakistan and fulfill our needs of meat and milk. So why there's a need to develop our own local animals? This is a common debate. So sustainable improvement is the key for long-term improvement of a country. So for sustainable development, we need to develop our own resources instead of relying on resources of others or importing from other countries. So sustainable and robust increase in local livestock production, and how we can achieve it, why we need to achieve it is, one is poverty alleviation. This is common thing. A lot of proportion of our population is living below poverty line. So we need to make sure that they are out of that poverty, and food security. And a recent is the climate change, because if we don't develop our own resources, climate is a danger for foreign breeds. If we import foreign breeds, they cannot withstand heat and diseases which are prevalent in this area. So we need to develop our own resources. Import-export, government is focused on reducing imports and increasing exports. So livestock is a big industry. We need to develop our own. Our feed resources are limited. Pakistan is a big country in terms of agriculture, but still we have limited feed for our local animals. So our local animals, they can live with limited feed resources, but if we import animals from other countries, they need a lot of concentrate, a lot of forages. So, and next is A1, A2 milk. You might have heard about A1, A1. A1 is a genotype. These cows produce bad milk. They have a genetic variant. This is a mutation, SNP mutation. So their milk consumption results in cancer and diabetes and many other dangerous diseases. And the most significant is lactose intolerance. So if you consume A1 cow's milk, you can get lactose intolerance, and a lot of problems comes with our infants. So our local breeds, they are producing A2 milk. Our buffalo is A2, our local dairy animals, they are A1, A2. So if we import animals, a lot of animals are already here. We have done a project with the collaboration of HEC, and we sampled animals and we tested them, and a lot of animals are A1. So we don't know whatever milk we are consuming, it is coming from A1 or A2. If milk is coming from local animals, this would be A2. So this is a typical our industry. Still, we are relying on buffalo. Buffalo is major dairy animal. Its contribution is 63% in our milk. National milk production. Cattle's contribution is 36%. And exotic animals, they are high producing animals, and commercial industry is relying on these animals, but still they are 1%. If we believe in this data, that is 1%, and if, for example, if they are more than this, they should not be more than 5%. So, we need to work on 63% and then 99% or 1%. Still, we have a lot of margin to work with our local animals. So, milk we are consuming is coming from our local buffalo and cattle, and only 1% proportion is of exotic meat. So, if genetic improvement works, it results in something. We can see this figure. This figure shows us that how genetic improvement, application of genetic technologies, they have contributed towards improvement of milk production. I have mentioned here only dairy cows. So our dairy cows below is Sahiwal cow, which is still giving around 2,000 kilograms in a year, and Holstein, which was giving 2,000 kilograms around 1950. So it's developed to 10,000 kilograms a year with this technology. So you can see the difference. A lot of difference is there, and we need to make up this difference. We can start from somewhere and we can reach at that level. So first thing was AI. This was the greatest intervention in our industry. Artificial insemination contributed a lot. Before this, we were doing natural mating. So with artificial insemination, we were able to disseminate the superior germ plants to a lot of cows, and it contributed towards improvement. And then around 1970, computers came and computer power came. So we started computerizing our data in America, North America. And they started recording milk in 1900 around. So they started computerizing and analyzing this data and identifying the best animals. And the cow which we are seeing right now, this was due to pedigree and performance recording. We were recording milk on cows, we were evaluating their data, and we were disseminating that knowledge. And latest is genomics. This has contributed a lot in robust improvement in milk production. So this is the pathway which Holstein took to reach 10,000 kilograms. And I would wish that we should start with our local breeds and take the same route and develop our breeds. So how genetic improvement and genomics work? Today we are focused on genomics, how genomics will contribute towards improvement. So firstly, we need to develop breeding objectives. This is, in simple terms, I would say we need to set the number of traits which we need to improve. For example, we need to improve daily kilograms of milk of a cow volume. Then we need to increase protein percentage, similarly fat percentage, and we need to improve their health and fertility also. So this is a first objective. Developing objective is a first step. And then we need to record data on pedigree and performance of those animals. This is the second step. And then comes data analysis. We would analyze that data and we would suggest which animals are best one. So this is the foundation for genetic selection. We will select genetically superior animals on the basis of genetics. And then artificial insemination and embryo transfer, these are reproductive technologies which would help us in disseminating their germ plants. And the upper is genomics. How genomics works? First step is development of a reference population. For example, in our dairy cattle case, to start with, we need to have 5,000 animals, 5,000 bulls plus cows, and we need to record their data on milk production. And similarly, we need their DNA information, which is called genotype. So the reference population is basically, this is the key, and this is the most costly and time-consuming component of genomic selection. If you do not have a reference population, you cannot implement genetic selection. A lot of people are talking about, they say, yes, we have genotype, for example, 20 animals, 50 animals, and we can apply genomics. So that is not based on facts. Basically, you need thousands of animals and thousands of animals with phenotype and genotype, and then you can develop a prediction equation. Here, statistics comes. A lot of biology people, they are afraid of mathematics and statistics, but here we need to develop this expertise in if we want to apply genomics. So when we have phenotype and genotype information, we will develop a prediction equation. And on the basis of that prediction equation, we can predict that what would be the contribution or production level of this animal in future. So once we have set the reference population, then with the help of this prediction equation, we can simply take a DNA of a newborn animal. For example, a cow, or a female calf, or a male calf is born today. We can take her or his DNA sample and we can genotype it, and then we can fit that genotype information in our prediction equation, and we can predict that what kind of production level this animal will have in his life. Right now, we are relying on real-time data. We need to wait at least 5 years before an animal gets its first production parameter. So with this technology, we would reduce that duration. We would get pretty much accurate estimate of its production level at birth. So this what we would get is predicted phenotype, which we call it genomic estimated breeding value. So right now in dairy industry, we are selecting bulls. So when a bull is born, right away we can take its DNA and we can, with the help of prediction equation, we can tell that what will be the production level of this bull, and we can predict genomic estimated breeding value. We can rank those bulls on the basis of that breeding value, and we can right away send those bulls for further dissemination. And the selection and culling decision would be made at birth instead of raising that bull up to 2 years, 3 years, and then wait for the records of his daughters, and then decide on its fate whether it is good for keeping or bad for keeping. So this technology helps us to select that bull at very, very early age. So what we need for the genomic selection is genotyping. Genotyping options are available. We have bovine SNP chip, 50,000 SNPs are available in this chip. SNPs are spread around whole genome of cattle. Cattle genome is about 3 billion base pairs. So they have developed, Illumina has developed a chip of 50,000 SNPs, which are correlated with performance rates of dairy animals. So this chip is available, but this chip is basically based on foreign breeds. No local breeds is in this 50K chip. Another option is 770K. This is high density chip. This is a bit costly, but it includes Bos indicus cattle. Our cattle is Bos indicus, and the chip is developed probably mostly on the basis of Bos taurus cattle. But in case of HD chip, we can use this chip for our local cattle to start with, to develop a reference population. And ideally, I would suggest, or I would like to have a custom chip for my own breeds. For example, a custom SNP chip for Sahiwal cattle, for our beef cattle breeds, for buffalo, which should be developed, and that would give us more accurate information of our local animals. Another option which I have not documented here, that is whole genome sequencing. That is more costly than even HD chip, and we need to have a lot of resources to store that data. For example, one sample would have 3 GB file, and that would be PET file and SNP file, and storing that data and evaluating that data in our system needs a lot of resources, and we need bioinformaticians to analyze that big data. So that would be costly option. We can start with the 770K chip, which includes both indicus information, and then we can do imputation. Imputation is a computer work. We can impute the missing SNPs which are not available in our local base, and we can generate information from that. So recently, I'm looking for genotyping my own animals, and we are facing this difficulty because nobody has ever done that, and I'm looking for how to handle that or how to get services from foreign countries. So we need to obviously we need to go to them for genotyping. No genotyping platform is available in Pakistan right now. I have contacted CAM, Center for Excellence in Molecular Biology, which is in Lahore, but they are doing small size genotyping or sequencing. This chip is not available with them. And with this technology, we need to have accurate identification of animals. We need to put RFID tags, which are the lifelong tags, and we can read those tags with the help of scanners. We have started this technology in our local animals, and this is required for precise and accurate identification of animals. Simulated benefits of genomics. In 2014, we wrote a paper based on information we had on our local animals and on the basis of genomics, and we simulated that how much benefit we would get from genomics. So this is a little bit complicated thing, but I would try to explain. These are 4 pathways: size of bulls, size of cows, dams of bulls, dams of cows. These are pathways of selection we call in genetics. And then selection accuracy, how much percentage of animal we would select, then intensity, and then accuracy, and generation interval, genetic standard deviation, and genetic mean per year, how much we would get. So with the help of traditional progeny testing program, we were having genetic gain of 86 kilograms per year. For example, we can increase 86 kilograms per year of milk production of each animal. But when we applied genomics in a simulation study in 2014, we got 256 kilograms per year. So this production level increased to almost 3 times. So we got 3 times genetic gain in performance and production, and this was the result of a half of genetic generation interval. So generation interval would be reduced with the help of genomics, and genetic gain would go 3 times higher than traditional selection, which we are doing right now. And then we did the same study on buffalo, because buffalo is our unique species. We are more relying on buffaloes than cattle as compared to international countries. They are relying on cattle. And here we got 2 times genetic gain was 2 times higher than traditional selection, and generation interval was reduced to half. And then we tried to estimate cost. This is conclusion. From 10 years, we were able to reduce the generation interval from 10 years in typical progeny testing program to around 3 years. And then response to selection was increased 2 times, and the cost of proving one bull was reduced to 88%. So we need to invest a lot of resources to prove a bull, whether it is used for AI and widespread use for farmers. So cost of genomics is not that much as compared to typical progeny testing program. But we are not experts of economics, so we might have done some mistake in this. But I would, our results were not very different from the international studies who have done the same comparative study of genomics versus progeny testing program. So how we can revamp this system in Pakistan? This is important because knowledge is there, technology is there. Now how we can implement this in our local resources, this is the challenge. I have done it, my studies in Canada, and everything is available there. We can easily get data, do analysis, and develop prediction equations. Thousands of animals data was with me, and I did my PhD study. But in Pakistan, recording data is a big challenge, and for this we will take the help of technology, RFID technology, and genomics. And we need a lot of collaborations to develop our local breeds. So collaboration is the key. We cannot develop, for example, I'm sitting in an ABG lab. I cannot develop my local resources, local breeds. I need help of all people, industry, farmers, academia, health sector. We developed our national center, and we developed collaboration with Livestock and Dairy Development Department, which is main stakeholder of our livestock sector in government. And then we contacted farmers, breed societies. We signed MOUs with them, and we are taking their data. We are taking data from Livestock and Dairy Development Department, and right now we are recording our own data. So we are in the phase of developing a reference population of 5,000 Sahiwal cattle, which we will use for genomic selection in Pakistan. This will be the first time. What our center will do, we will develop trained manpower. To the best of my knowledge, there would be less than 20 PhDs in Pakistan in animal breeding and genetics. So trained human manpower is required. We have started our MPhil and PhD programs there to develop students, postgrad and MPhil and PhD students who are expert of genomics. Recently, one of our students graduated in livestock genomics. So we did 2 theses in livestock genomics. We did this technology, applied this technology in both. And all local report breeds are involved. And then comes sustainability and funds. How we can achieve this goal? We can realize we need funds and sustainability. We have funds from HEC, and then Livestock and Dairy Development Department will contribute in this. They are contributing. And these societies, they are a little bit weaker component of our, because these societies are very limited region. They are not that much advanced as they are in USA and Canada. So we need to work with these societies to develop them, and they can also contribute funds and their expertise to apply genomic selection in Pakistan. Thank you very much for your time. If you have any questions.
5 questions, one by one.
Thank you very much, Dr. Bilal. I think you did very great. It's a nice talk. I just want to clarify one thing regarding, you mentioned A1 and A2, if you can explain a bit. Like, is it, which one is better? And what basis you can say which one is better? One thing. And second question is regarding that we also have, you mentioned 1%, we have an exotic animal. So when we're importing some kind of animal from other countries, so do we have any kind of a criteria where we can check either they have what kind of genomics they have? Because I found out that either this is a rumor or maybe it's a truth, but it depends upon the also being a Muslim, we have a concern like halal or haram. So do they have some kind of, you can say in terms of GMOs, do they have incorporated some kind of genes from other animals that are not halal? So if you can explain a bit about it.
So A1, A2 is a common slogan in New Zealand and in Canada and in USA. They are labeling milk with whether it is A1, A1 or A2, A2. So A2, A2 milk is desirable, it is costlier, and then the bulls available in the world for dairy cattle, for example, when you read a size summary of bull, they give the result whether it is A1, A1 or A2, A2. There could be 3 types of animals, whether A1 homozygous. We are 100% sure that they are going to produce bad milk, and then heterozygous A1, A2. Here we are also clear that some proportion of that milk would be dangerous because they are 50% lethal, and A2, A2 they are good animals. So if a bull is A2, A2, we have written to the Livestock and Dairy Development Department, and because they do international tender when they import semen or bulls, so they have written now that it should be A2, A2 bull. I don't know whether they are complying to it or not, but we have managed to compel them that they should set this criteria. So this is regarding A1, and what was your second question? I missed that.
This is regarding basically, just like there was some kind of certification, whether they are not inserting any kind of foreign genes, or in terms of breeding program.
Yeah, we are not importing any genetically modified animals, because genome editing or genetical modification, there is a difference. Genomics is just selection. We are not modifying any gene. We are just trying to identify genes that are at an earlier stage than at a later stage. So this is just selection. We are not changing any. So I don't think we are that much concerned on that. Our end mathematics mission is Islamabad. They deal with import and export. And I don't have any exact knowledge of what criteria they set for import and export. There are a lot of things. Any other question? Right.
Informative talk. Thank you so much. I just want to ask two small questions. Number one, you talk about the good milk, bad milk, A1, and A2. What is the difference between these two, either in the composition or why it is bad? Secondly, in the slide number 5, you gave the data related to the cattle. Is there any data of buffaloes available also? In slide number 5, you have
Yes, I think I mentioned Buffalo was there.
from the comparison between the milk production, feeding and
Yes, no, Buffalo's level is around, it's close to Sahiwal cattle. It could be 200.
it isn't right as
No, because we did not apply any selection strategy, so it should not, its average is around 2200 kilograms per year. Buffalo is same, yes. Milk composition, that is a beta casomorphin, which is on chromosome, that is a mutation. So that is excreted in the milk of A1 cow. So that gives lactose intolerance and diabetes and certain forms of cancer. So its studies a little bit controversial because Americans, they don't like this studies because this is most related to New Zealand and Australia. They are labeling A2 corporation is available in the world. They are labeling this. A lot of studies are available, but they don't like it because they have a big breed and their 50% Holstein, American Holstein is A1. So a lot of high allele frequency is available with them, so they would never propagate that.
Dr. Bilal, what is the status of progeny testing program for buffalo? And secondly, is it possible to do the genomic studies even if we do not have the proper record of the genotype?
Well, to second question, that is big no. We cannot do that if you do not have reliable data. We cannot implement any form of genomic selection or any strategy. Data recording setup is available with Livestock and Dairy Development Department, and they are doing from 1960s. But question is the reliability of that data.
So, what is the current status of that program?
Current status, I have recently visited the Buffalo Research Institute and we inspected their system, but they did not provide us any data. They do not have a computer lab, and they have the director had a laptop and he said our data is here. So I suggested them to buy one more because if that laptop is stolen, then Buffalo's data will be.
Actually, that program is not testing and nothing is available now.
Yeah, that's true.
तो मुझे चाहिए था मुझे सिर्फ 17 एनिमल का मिला है
That's true.
only 17 buffalo.
Sir.
Yes.
So I have a question, thank you very much for nice talk. I have a question that in Punjab, there is a department of breed improvement in livestock department, and they have developed the breeding policy. Either the things you are talking about or you are revamping the things, either they are being included in that breeding program because of massive cross breeding. We have been here for this problem, like A2, A1 or something like that, with different type of milk. Either that what you are talking right now is being included in that breeding policy or not.
Yes, we are working with them, but problem with L&D is that they do not have a lot of experts in this subject. So it is hard to convince them. Their secretary is a non-technical person. So that is one limitation. But still we are including them. We are doing a lot of meetings with them. We have MOU with Livestock and Dairy Development Department. Directorate of Breeding Improvement is with us. They have a lot of semen production units which they are running under their control. But they are not applying any scientific selection right now.
But I have heard that they have developed the different conservation centers like RCTSC and Government Research Institute, but they are relying more on the animals, they are infecting the animals on the basis of the mass action and something like that, but they are having the concern like this because these are emerging issues and they are being the global issues. And importantly, if we will not consider these issues, so what will be the future of all these kind of things which we are doing right now with the university, if they are not being implemented, if they are not being prioritized at level of government or at level of government?
Yeah, so we will do our part and they are supposed to do their part. If they don't do this, obviously nothing would happen. Recently I talked with the Director of Breed Improvement and there are 17 private SPUs and on 15 SPUs they could not able to trace the pedigree of those bulls which they are using for insemination. So when pedigree is not available, then performance or other things we cannot expect what it would be. So that is random semen which our farmer is using and we don't see any form of improvement. We should not expect anything from a random semen.
Because in the past we have destroyed a lot more semen from the SPUs.
Yeah, that was the reason.
Having the from the tested bulls or the proven bulls, I think so. I have seen with my own eyes that only 2 bulls were they have been proven in case of buffalo. I don't know if they figure about the cattle. But the thing important is that they are being the point from where you can bring a lot of change, rather in the province, rather. But only in Punjab I have seen the breeding policies there, breeding act is there, but you must be at an appropriate forum to implement it on there around the country, so that the things you are talking about that can be implemented in the entire country.
Okay, sir. Thank you.
Last question. Yes.
Thank you, Dr. Bilal. I just appreciate the comments of Dr. Imtiaz because there is an issue of implementation. Could it be possible through our center we maintain a nuclear third at university level so that we can be able to provide authentic genetic material to the farmer?
Yes, sir. We have this component in our project and we will develop a semen production unit in our food research farm. And we will develop a semen processing lab here, and we would try to be model for them to implement these technologies. And we have this component.
Whenever we have a workshop, the farmers they always ask from where we can get the pure animal. Ultimately, when there is no implementation of all the things, so if we are able to do this at a university level or have a nuclear service, that would be a very great achievement, particularly for the development of this country. Thank you.
Thank you, sir.
Thank you, Dr. Bilal, for a wonderful speech. Now I request all of the questions to have a few words on conclusion of the session.
Thank you very much, Dr. Bilal, very informative and nice presentation. And we invite your presentation. And thank you, Dr. Manpower and his team to organize this seminar. You already mentioned about this advanced technologies and have been developing a center at this campus, which is a contribution towards this university and our country. And as you mentioned, we know So without now technology implementation, development and implementation, we cannot progress in agriculture, in animal science, veterinary science. So this is a very good learning for us that we have this facility, or we will have this facility at our campus, and for capacity building of highly qualified personnel, graduate students, and PhD, and our faculty, and that is one part is technology, and we are, as you mentioned, in North America, especially in Canada, we are far behind from those countries, but and we learn, I think, lessons as some of our colleagues mentioned here. So we can improve our system. But we are in a system that we have to collaborate with Punjab government, institutions, and federal government. There are some limitations, but as Dr. Khan said, we have our own part. So number one is training our students and faculty in this new research area, and new labs. Hopefully, we will have these physical labs in during this year, and their animal that shed at home farm. So and veterinary faculty, D and his team have been working with this Dr. Wayne and Bilal, and we now started weekly, bi-weekly meetings with other supporting partners to grab this facility as soon as possible. So without these innovative advanced technologies, and I think trend is there now, some progressive farmers have started importing those gold and gold. But without, and again, the issue is some of them contacted me that we need some skilled training farm managers for this dairy farm, or this for cattle for meat. We don't have many, especially in this area, artificial insemination and new transfer. And again, not only this in livestock, I have been asking our faculty for agriculture production systems also. That data, reliable data is extremely important. So otherwise, हमारे सीनियर प्रोफेसर कहते थे कि खाना खावा भी जाकर तो माफी हो जाएगी, डेटा में गलती हुई तो माफी नहीं होगी, क्योंकि this and our poor people and especially in this area of meat and milk, you cannot find pure milk now. Big challenge. So if we have correct data, so then we can predict with these advanced technologies, models for future generation of animals and milk production. So it is our duty. Religiously, we have to train our students for reliable data collection and technicians. And I encourage all our faculty, please go with your students. If you need any kind of transportation, any kind of facility, so the directors are there, and we have vehicles and get them go with the students. That is the first step. Then we need to learn about these new techniques. Some of our faculty are facilities now, I mentioned many times for agriculture, for this animal sciences, mega projects that we are needed in those techniques and technologies needed at agriculture university. I don't think Kasaba University or other universities have those kind of facilities, industrial biotechnology, and all will be IS certified advanced lab and proceed agriculture, digital agriculture. And we, in this livestock breeding and genomics, and they will have this main data center here and remotely collecting all the information, all those new IT applications and sensors and control system. We are collaborating with Sparco, and now we will work with the Pakistan Air Force to develop and implement these. We have opportunities now. Challenges, we have challenges, but we have also opportunities. Now, I think it's a very good step. We are sharing this knowledge with each other. Not only with this lifestyle genomics, but for example, this IT application, have been working closely with Dr. Khan, and Dr. Bilal with this project. So the technology is almost similar, but like data center to collect data from different centers for animals and agriculture crops. So will be the same, technology will be the same. Some of the sensors can be used for both purposes, but we need to learn. So, and one sensor that can be used for agriculture production system, and the same can be used in dairy and poultry. So, we have IT department, we have economics for Dr. Bilal mentioned that we are not, and we have economics department here, so sociology, we have business management, and then engineering department. So we need to integrate and collaborate with each other. And I have seen posters yesterday that extension department, they have very nice topics, digital farming, but the faculty and student don't know that we have a mega project in Precision Digital Agriculture. We are not collaborating. So I always actually meeting in the department. So if we collaborate with each other, and the extension department is like just like I heard that how we can disseminate our results and transfer technology. That's what we have extension department. So we need strong collaboration, and biotechnology, those techniques are similar in animal science and agriculture science mostly. So we have all those, and funds are available now, and facilities are there, vehicle transport, no issue there, manpower, no issue, and labs, we have funding to repair our equipment. So if you have somebody in your lab, equipment not working, you can contact Dr. Javeed or people, they will help you. So, and secondly, please encourage your graduate students to participate in these seminars more and more. They can learn more about how to even present or how to prepare this PowerPoint presentation. There will be workshop in the calendar, but it's a very good opportunity. So Dr. Bilal, he graduated from the university, very prestigious university, and these kind of presentation techniques, and so each and every of our colleague has some kind of skills. So graduate students, our main goal is to train our students. So each student, single student is an institution. So please, so I again encourage to spend more time with your students. They are future our leaders, and if we train सबसे बड़ी तब्दीली यही है। तो ये जो 3 दिन का चिल्ला भी जरूरी है, लेकिन ये चिल्ला क्योंकि लोग जो है वो 2 टाइम की रोटी नहीं खा सकते, ये जो मजदूर काम कर रहे हैं, ये हकीकत है कि वो और अगर हम ये मिल्क और मीट की प्रोडक्टिविटी बढ़ा सके सारे मिल के, तो ये बहुत बड़ा सवाल होगा। तो ये इस पे गौर करें। तब्दील को मैं उनके खिलाफ नहीं, तो नमाज तब्दील हर चीज जरूरी है, लेकिन ये सबसे जरूरी है, हमारा फर्ज है, क्योंकि अल्लाह ताला ने हमें ये मौका दिया। और so we are very few, maybe 0.1% PhD in Pakistan, and so that is our duty and responsibility. तो अल्लाह ताला ने ये भी पूछना है कि आपका फर्ज था, ये भी फर्ज है। तो और मुझे सबसे ज्यादा तो मैंने ये जिम्मेदारी कबूल कर ली, तो मुझे भी चाहिए और आप
Thank you very much for the Vice Chancellor. मेरी लास्ट की रिक्वेस्ट है रिसर्च कोऑर्डिनेटर से, चेयरमैन हजरात से, फैकल्टी मेंबर से, डीन डायरेक्टर से कि अपने स्टूडेंट्स को, अपनी फैकल्टी को रिक्वेस्ट करें, उनको इनकरेज करें टू जॉइन दिस स्टैंडर्ड। क्योंकि हमने ये एक्टिविटी स्टार्ट की है, ये एक्टिविटी आप लोगों की कॉपरेशन के साथ हो सकती है, अकेला एडवांस स्टडीज का डिपार्टमेंट इसको नहीं रन कर सकता। तो जैसे आज मैंने वाटरी की थी प्रेजेंटेशन, मैंने जोलॉजी से, मैडम केमिस्ट्री से, एंथ्रोपोलॉजी वाले से रिक्वेस्ट की कि आप लोगों का रेलेवेंट फील्ड है। इसी तरह जो स्पीकर मुतलका होते हैं वो मुझे रेलेवेंट फील्ड बता दिया करें। मैं भी रिक्वेस्ट कर दिया करूँगा मुतलका चेयरमैन हजरात, 3 हजरात रिसर्च कोऑर्डिनेटर, वो भी मेक श्योर करें कि रिसर्च स्टूडेंट्स और फैकल्टी पार्टिसिपेट करें। अभी स्टूडेंट नहीं आए, मार्कशीटों से स्टूडेंट्स और भी आ जाएंगे, उनसे भी हम उनको भी गाइड करेंगे। अभी आप लोग जो अवेलेबल हैं वो मैक्सिमम आप लोग पार्टिसिपेट किया करें। थैंक यू वेरी मच फॉर योर टाइम। थैंक यू डॉक्टर बिलाल फॉर योर प्रेजेंटेशन। ठीक है, आप लोग कोई दिक्कत से